to all of you and welcome staff and students and guests, as well as staff from other faculties at the University of Cape Town. And today is a very special day. Aside from this very special seminar that we are hosting, we are also honored and very excited to have our new dean in the Faculty of Health Sciences who starts his work today. So I would like to call upon our dean, ex-dean, Professor Sophia Kisting-Cairncross, Professor Lundy Braun, as one of the through one of these functions that we are hosting today.
Thanks, Mohamed. Thank you very much. It's wonderful to have this particular very important seminar today, on the day that all the deputy deans can get to it, and on the day that our new dean has arrived. So we've taken an opportunity wherever we have to say welcome to Professor Braun. We are not going to ask him to speak, but we're delighted that he's here particularly for the seminar, and to welcome the guests and the speakers, and we look forward to hearing the debate. Thank you.
I also want to say that Professor Braun, DBC, who is responsible for transformation, sent his apologies he wasn't able to be here today since he had another competing engagement. So what I wanted to share with you, I'm not going to talk very long because we have an eminent group of people to speak to us, but just to share with you for 2 minutes some of the ideas behind this particular seminar. And to say that transformation and equity are one of the 6 goals of the Faculty of Health Sciences, which is reflected in the mission statement of the faculty. It commits the faculty to ensure that transformation promotes the development of a faculty of equity, reflecting the diversity of society. At the university level, the ongoing transformation of UCT towards a non-racial Is that better? Okay, all right. Okay, so transformation and equity, as I indicated, is one of the 6 goals of the Faculty of Health Sciences, and this is reflected in the mission statement. It commits the faculty to ensure that transformation promotes the development of a faculty of equity, reflecting the diversity in society. At the university level, the ongoing transformation of UCT towards non-racialism, encompassing redress, diversity, inclusiveness, and the recognition of African voices is one of the university's 6 strategic goals. So as part of our ongoing education program on transformation in our School of Public Health and Family Medicine, we decided to host the seminar and panel discussion on knowledge, equity, and health in post-Apartheid South Africa. What's race got to do with it? In order to move us closer towards this goal, which cannot be reduced merely to numbers and institutional culture. Knowledge production is an integral part of what we at the higher education institutions engage in through our research and teaching and training activities, and through providing health care to the population. The School of Public Health and Family Medicine has been mindful and deliberate in finding innovative and inclusive ways to redress at university level some of the problems created by the apartheid system, so as to contribute towards transformation and also other initiatives that occur at the university. Despite these efforts, there continues to be formal and anecdotal reports from some of our students about concerns with regard to discrimination and inequality that they experience through their teaching and learning activities. So with South Africa's long and dedicated struggle for the attainment of universal human rights for all, And because of our distinct history of apartheid and inequality, we continue to contribute in finding innovative ways to overcome these challenges, collectively by drawing on the inputs of members in this faculty and abroad. Ultimately, we would like each and every student to have the opportunity to reach their full human potential in an enabling environment in their quest for knowledge production, transmission and dissemination, and providing quality healthcare. For our staff, we aspire to produce excellent role models in the faculty that our students can emulate. So the school is very excited and honored to have with us a panel to discuss this important topic. We have Lundy Braun from Brown University in the USA. We have Raj Ramesar, Head of Human Genetics in the faculty, Glenda Wildschut, Director of the Transformation Office, and Sophia Kisting, who is the former Director of the ILO Global Program on HIV and AIDS, who was based in Geneva. So that's all that I want to say as a context. What we're going to do now is have our keynote address by Lundy, and then we will have some questions that she could take following her presentation. And then we'll have our other panel members who will join her to respond to her input as well as to draw on other aspects relating to this particular topic. Lundy is a professor of pathology and laboratory medicine and Africana studies at Brown University in the US. Her research takes an interdisciplinary approach to analyzing the structural causes of health and inequality. Lundy's current research focuses on the history of the use and understanding of race in public health and medicine, especially as it relates to the racialization of lung capacity measurements. Her book, entitled Breathing Race into the Machine, The Springing Career of the Spirometer from Plantation to Genomics, will be published soon, and we look forward to reading that particular work. So over to you and welcome, Lundy.
First of all, let me begin by expressing my thanks to the Faculty of Health Sciences. I'm really very happy to be here again, and particularly to participate in a panel discussion about a topic as important as this. From my perspective, there's nothing like dialogue, and we need to keep the dialogue open, not let it close down. In the many years I've been coming to South Africa, I found the discussions with colleagues here of absolutely critical importance to my thinking, both about my research on race in medicine, but also about society as more broadly, and of course that's what it's all about. Whatever perspective we take on the question of race in medicine, those perspectives are impoverished by an exclusive focus on a single national context. So I've tried to draw some points about what looking at some of these issues transnationally can help us see. There's great enthusiasm right now in universities of the North for internationalization, and I'm sure you're well aware of that. Though partially well-intentioned, what this actually will mean in practice is unclear, and there are some worrisome signs, and I'll mention some of these today. Given the overabundance of resources in the US, we must be alert to the very real dangers of globalization of US ideas, techniques, practices, habits of mind, and notions of race. This is particularly problematic in the case of science, where under the guise of objectivity, cutting-edge technologies, or standardization projects, globalization seems to be neutral. The topic of this panel is broad with many different dimensions. My remarks will draw on my research to focus on one tiny aspect of this issue, and we can put it all together, some of it together, in the panel discussion. So what I want to focus on is the use of racial categories in the production of scientific knowledge centered on the causes and prevention of health inequality and under the rubric of public health and certain aspects of medicine, and I'll use the term biomedicine to refer to it too. So I've been very interested in my research, what knowledge is being produced, but also what knowledge is not being produced because of the ways we think about that knowledge. So for example, race. To what extent has race, does the lens of race actually inhibit our thinking about other causes and prevention of disease? The US and South Africa share the dubious distinction of particularly troubled history when it comes to questions of racial oppression and discrimination. But that said, I think one thread I want to keep through this talk is that while our history, while we share much about our histories are also really different. And so the mere importation of US ideas or importation of South African ideas, as happened in the history of lung capacity measurements, we have to be very cautious about. But this history is both a social history, a cultural history, political one, but it's also scientific. And the science can be much harder to see because we think generally think about science as a neutral endeavor. The history of science, in fact, shows us that science and biomedicine merged together with race, colonialism, and imperialism, and that they are inextricably intertwined. This science has often been called somewhat dismissively scientific racism, but in fact it was normal, well-respected, mainstream science that produced the concept of race. And the science has always been global and persists. Because of our shared history, the US and South Africa historically have looked to each other to affirm race as deep difference. While much of my work is centered on the US context, I've been interested in these global exchanges of knowledge. For a variety of reasons that deserve more careful explanation than I can give today, we're at a moment when the idea of race as genetic difference is being revitalized globally. This was not a completely predictable outcome of the drafting of the Human Genome Project in 2000. In fact, when the draft was announced, the dominant discourse at that time was we were all on which human genome was going to show our common humanity. But very quickly, a flurry of studies, many framed as health disparities research, and promoted by activists from groups representing women and minority groups in the US began looking at genes as causes of racial disparities in disease. And so I want to make that important distinction about what happens when we look at genes as causes of racial disparities and racial and ethnic disparities in disease. A direct result of the NIH, of this activism, was that NIH mandated the use of race for all funding applications. And this has been detailed quite nicely in a book by Stephen Epstein. The search for the causes and treatment for racial disparities in disease is often referred to now as race-based medicine. It became very high profile with the battle over Bedevilled. I'm not sure how much that hit South Africa, but it was a drug for congestive heart failure, which was approved by the FDA for blacks only. It was only tested in blacks. Much has been written about that. I won't go into detail, belabor the point here, but the use of race on the part of the company, Nitromed, was merely to get patent protection for the drug. It was actually a combination of generic drugs that were much, much cheaper. Even though this drug hasn't panned out, the concept of race-based medicine has become deeply entrenched in U.S. biomedicine. The multinational pharmaceutical industry and its quest for profits has played a major role both in the revitalization of race as genetic and this is a key point I want to make, in obscuring the social causes of disease. Ian Whitmarsh, Ian Whitmarsh has demonstrated the complexity of internationalization in his book, Biomedical Ambiguity, which is an ethnographic study of genetic research in Barbados. Here he gives us a sobering insight into what internationalism can mean for under-resourced nations. The questions of genetic researchers from the US and the UK, the multinational pharmaceutical industry, and the healthcare challenges of a resource-poor country came together to establish Barbados as a laboratory for genetic research, and in the process stabilizing notions of race that fit with US questions, UK, US questions in ways that had absolutely nothing to do with the identities of people of Barbados. The entire system of healthcare is now shaped by genetics and pharmaceutical research and interventions, and local physicians, government officials, other partners, and sometimes patients have bought into this, but not without contestation. Coincident with the proliferation of genetic research, a rich literature in sociology, medical anthropology, history, questions the conceptualization, methods, and designs of these studies, yet scientists almost never read these books, and so there's very little dialogue. Indeed, biomedical journals will generally not publish these sorts of analyses, or only very watered-down versions of them. Words like racism can never appear. But academic presses, ironically, can't swoop up the books as fast. They're just fast enough. There's an absolutely important, troubling, and fascinating disconnect between research going on in different spheres. One explanation for this revitalization of a purportedly genetic basis of race is that, of course, race is essential difference never went away. Sometimes prominent and other times lurking beneath the surface, race was, as Troy Duster writes, buried alive. Now it is rooted in genes or SNPs, while explanatory frameworks have historically oscillated between environmental, behavioral, and innate factors. An undercurrent of intrinsic difference, especially between people labeled black or people labeled white, has persisted for at least a century and a half, if not more. I want to make just one quick point before giving a very brief overview of my research, and that is to look at the census categories, which are the social formations that are typically used in biomedical research in the US. And these census categories are getting globalized. So Melissa Nobles, some years ago, documented what groups were included in the census categories. And what she finds, and illustrated on this slide, is that every single decade, every single US census from 1790 changed the categories. And the criteria for who was what changed. What's interesting about this, and I'll just point out, is that we have what you call the one-drop rule in the US, where one drop of white makes you black, whatever that drop is. But I just realized, actually, in preparing this talk, when that happened. And that happened in 1930 census, when more complicated ideas of what might be black were erased. You see the mulatto category disappeared, the octoroon, and as offensive as they are, what they did was trouble an absolute black-white divide, and that black-white divide emerged in 1930. Brazil has an entirely different approach to the census, in that they use color. And then, of course, as you well know, South Africa has a whole other approach to racial classification, and it's changed many, many times. My point is that it's hard to argue that these categories are natural, even though they are being argued as good enough by genetic researchers in the US. But you would have to first decide who really nailed it. Which census, US and South Africa are among the 15% of countries in the world that even use race. So whose census is natural? Okay, so let me now just, even Wikipedia recognizes that there's a problem with the census categories. Okay, let me now turn to one tiny piece of the story, and that is the history of the racialization of lung capacity measurements, to illustrate how racial difference became entrenched through ordinary scientific practices, such that it took on the character of objective truth, and such that it indeed linked class spirometer, an instrument that measures lung function that is well known to many of you here, is used worldwide to diagnose and manage respiratory disease. The spirometer on the top is from my doctor's office, the much more modern looking spirometer on the bottom is from South Africa. Based on a centuries-old scientific consensus that racial and ethnic groups vary in the capacity of their lungs, commercially available spirometers correct or adjust for race in one of two ways: by using a scaling factor for non-whites or by applying population-specific norms, both of which can be programmed into the instrument. So basically, I've done lots of interviews, and the majority of people don't even know the race correcting when they're using the spirometer. The consensus view on racial difference is based on a fundamental methodological and conceptual problem, and that is that the majority of studies on racial difference did not define race and or ethnicity. Despite this failure, this is the result of a very onerous systematic review of 226 studies from 1922 to 2008. Despite this failure, investigators throughout the 20th and 21st centuries have consistently compared lung function in various racial and ethnic groups. And this is another key point, invoked inherent or anthropometric factors as explanations for observed differences. So what's important here is how race is being conceptualized and then how difference is being explained. And if you look just at the pink and blue, you can see that there's, the numbers of studies in the 20s were much smaller. So, but you can see there's a certain consistency in inherent and anthropometric factors. But there's an increase in the 2000s. There's no explanation, and that has its own problems. There's a very, very small literature that looks at socioeconomic factors in lung function. And so the point I want to make is that race is the almost exclusive lens through which variability in lung function has been studied. As far as I can tell, the notion that black and white lungs differ dates to the early years of the American Republic, when Thomas Jefferson, writing in his notes on the state of Virginia, posited many visible distinctions between slaves and whites, including a difference in the pulmonary apparatus, and that's a quote. Jefferson's musings would receive an empirical foundation 6 decades later when pro-slavery, these are two New England Journal of Medicine that just sort of layer new genomic technologies onto the old race-based model. So, pro-slavery plantation physician Samuel Cartwright used the spirometer, he actually built one, this is in 1851, to compare lung capacity in blacks and whites. Drawing on Jefferson's interpretive framework, Cartwright reported that race, together with other factors, affected lung capacity, and he quantified the deficit at 20%. Thus, we think that the idea of difference is simply a product of the backward South after Cartwright, which is looking pretty common in the US to just look at the South as backward. Lung capacity research shifted to the northern US when the esteemed astronomer Benjamin Apthorpe through Gould organized a large anthropometric study of Union soldiers at the end of the Civil War. Gould's summary of the study included a chapter devoted to lung capacity measurements in black and white soldiers. Gould's work was then picked up by none other than Charles Darwin, who included lung capacity together with craniometry as indicators of racial difference in his Descent of Man. The racism embedded in Gould's ostensibly neutral scientific language became more explicit when his data was marshaled by Frederick Hoffman in 1896 in a really vicious, almost impossible to read racist diatribe, race traits and tendency of the American Negro. Making broader claims than Darwin, and this man was very important to occupational health, so it's a really interesting history here to unpack. Making even broader claims than Darwin, Hoffman wrote that the smaller lung capacity of the colored race is in fact proof of an inferior physical organism. According to Hoffman, blacks were therefore unfit for freedom, so he made very large social policy policy conclusions based on lung capacity measurements. Leading African-American intellectuals W. Du Bois and Kelly Miller wrote trenchant critiques of Hoffman's arguments, yet their really compelling analyses failed to alter the scientific discourse on the meaning of lung capacity measurements. Interestingly, only Gould's purportedly neutral observations, stripped of context and explanation, not Hoffman or Cartwright's more strident narratives, would be cited in future scientific literature, including the article in the Journal of Medicine in 2010. In the 1920s, at a time when eugenic policies rooted in hereditarianism were popular in the US, the study of genetics of race converged with the emerging field of human genetics, and racial difference in lung function became more legibly scientific. The prestigious Journal of the American Medical Association published the first set of spirometry-based lung function standards in 1922. This difference, the author speculated, could be a racial factor. Basically, by the mid-1920s, the evidence was considered irrefutable. Not much happened in this field until the 1960s, when comparative research flourished internationally, and South Africa was important in this flourishing. While the U.S. remained the major source of studies comparing lung function in blacks and whites during this period, they were joined in this work by researchers from other countries such as apartheid South Africa, the UK, China, and India. The most highly cited remained those studying individuals determined to be black. The U.S. and South Africa, and I've been following citations, patterns of this literature. The U.S. and South Africa were particularly important because these two countries had resident populations of blacks and whites. And so what that meant is that they could do controlled studies, the gold standard of scientific research. In this period, in the 1960s, and if we think of the 1960s internationally, there was a lot going on that should have troubled this narrative. And it did to a little extent. There were a wider range of factors such as respiratory infections, tobacco smoke pollution, but the dominant frame of intrinsic difference between basically blacks and whites persisted throughout the 1960s in the US and elsewhere in the world. During the 1970s, the very idea of innate racial difference in lung function gained an even deeper empirical foundation, even though the research, as I said, failed to define race and ethnicity. And this was reinforced in a transnational collaboration by Charles Rossiter from the Pneumoconiosis Research Unit in South Wales and Hans Weil from Tulane Medical School. And they came up with the scaling factor that is well known now, or also called a correction factor, of 13.2%, and was applied to blacks only. No attention was made to the various meanings of black worldwide, apartheid categories in South Africa, changing categories in the US, and so on. And blackness was assumed to reflect deep biological difference. In the history of this practice, South Africans, Johnny Myers and D.L. White and others were, as far as I can tell, the only one to interrupt the dominant frame in the 1980s and 90s by questioning the practice of race correction. I think this speaks to the importance of context and how we think about our science. So while South Africa produced much cited studies that influenced the race-based model, it also produced the only contestation of the assumptions about race since Du Bois and Kelly Miller. The South African work, however, received little attention outside of South Africa, whereas the other studies that reinforced the race-based model were much cited. Some of them were much cited, not all of them. A systematic review of lung function studies on the African and African American populations, written by Neil White and colleagues many years ago, gives tantalizing clues as to what questions we might not be asking. And I want us to think about that in the discussion, hopefully. First, he found that, surprisingly, there was great heterogeneity among both African and European populations that were masked when you just Moreover, and this is really very interesting, and as far as I know, hasn't been followed up, but there were pronounced secular trends, and the slide on the top is from his paper, shows the secular trends in populations in Africa in lung capacity, and the slide at the bottom shows the US. And what you can see is that lung capacity improved on the continent of Africa, whereas in the US it declined. So the secular trends went from opposite directions. Again, raising questions about the naturalness of difference. So let me conclude. The categories we use to frame biomedical research on race matters. And these categories have histories that we can't ignore. In his lovely book, Silencing the Past, the anthropologist and historian of Haiti, Michel Roth-Triot, tells us that, quote, "We are never so steeped in the past as when we pretend not to be." Power, Triot continues, makes certain narratives possible and silences other narratives. Looking at patterns of disease strictly through the lens of race or ethnicity not only reifies race and ethnicity as genetically circumscribed categories, but it also obscures the social and structural dimensions of health and disease. Nearly 2 centuries of work on race and lung capacity has led to relatively little understanding about what causes lower lung capacity or the variability and its relevance to disease. There's really compelling, I think this is the best thing that has been published in years and years and years. Bruce Link and Joe Phelan, and there's a student at Columbia Public Health School, demonstrate empirically in a variety of different ways that explanation about genetic difference coming from scientific studies fosters racism among the public. And the situation's getting worse with younger researchers more wedded to genetic models of disease than older ones, primary care practitioners getting drawn into this in ways that they're very uncomfortable with, but nonetheless find it very hard to review. So only by learning to see how the past lives in the present and identifying the processes by which this past is sustained, and that's, my plug for history, can we hope to be able to ask new questions. We won't get answers to questions we don't ask. Thank you very much.
So we have some opportunity for people to ask questions in response to the presentation. So I have a question. Despite the fact that we are 20 years into democracy, the use of racial classification in this country is also continues to be used, but for different reasons. You have argued that the use of race has been used to blur, for example, the social inequalities. The reverse argument has been that the use of race in this country is being used to deal with issues on how far the government has gone so far as addressing the inequalities as a result of apartheid. How would you respond to such a
Okay, well that's of course the big question. I mean, to be honest, I'm somewhat reticent to take an absolute position on the situation in South Africa, in part because every single time I come, I learn more about how different, actually, the use of race in South Africa and the U.S. is. So I have a really strong position about the use of race in genetic research. I'm actually troubled by a lot of the social research that happens. And so I'm emphasizing one thing here, but I think a lot of the social research that is going on, everywhere is voyeuristic, and I think it's people are not thinking about what the real question is and whether we should subject people to some of the kinds of studies we're doing. So, and the social often is framed in terms of individual behaviors rather than structure. So I think the use in medicine is complicated. I think in the clinic, there's actually no view that race, and I've thought this through carefully, and I have a paper that I published with a bunch of people, that in the clinic, it's of no use. That it gives you a shorthand that's actually going to lead you way down problematic categories. So, for example, in the US, if someone you think might be black, and you're making all sorts of assumptions with that, that people are asking questions about, I have to say, but that they have hypertension. What is that going to tell you? And we should check everybody's blood pressure, you know, and then if somebody has hypertension, then you deal with it then as an individual. So I think in the clinic situation, it is being used as a shorthand, it's being defended as a shorthand, but it's a shorthand for actually not taking time to talk to patients. I think in many areas of research, it should not be used. I think the area of redress is where I think it's quite complicated. I'm increasingly stunned by the ways, as much as I've read and talked to people, it's so imperfect because I didn't live it, and the ways in which apartheid racial classifications really penetrated the crevices of life. And in fact, that is, although US is very racialized, I mean, the murder of Trayvon Martin is a really good example of how deeply racialized we are, that the bureaucratic structure, we don't have to fill in race for anything. I mean, it's assumed, it can be asked, I mean, for sure race is operating because people are being operating on those assumptions, but there is something different in the bureaucratic edifice that was built here. And so the extent to which it is used does in fact perpetuate, I mean, the real risk is perpetuating the past. So that's sort of a convoluted answer to.
Other questions?
Looking historically, it seems like that, you're discussing kind of a recurring pattern where kind of essentialized racial thinking keeps emerging in different forms, different scientific kind of packaging of it, but this is still kind of gone. Right? And the fact that many studies don't define race demonstrates that also it's almost like a negative example because it doesn't say what people are actually thinking, how they're actually constructing it. Just points to essentialize an unasked question.
Right.
So with that as a kind of context, what I was wondering is how you have seen, it struck me that you started off talking about health disparities researchers, who as it's kind of framed in the States, would come to a very different political position. And wouldn't, I would imagine.
What do you mean?
When they would come from a progressive, liberal left kind of school of politics, when trying to,
Loosely, very loosely. Right.
But what I'm wondering about is this tension between the impulse to it's a kind of follow-on, I guess, to Tom's question, the impulse to try to name and address racial disparities or gender disparities. How do people who are working in that field in the States try to balance it kind of thread back to where you did this? Because so much goes unspoken.
Exactly. I think that's really important because, writing this paper for a scientific journal was excruciating for me because of the things I couldn't say. I mean, in fact, people weren't using a definition of race, they were using common sense notions. But interestingly enough, for a scientific paper, they didn't have to define it, which is, that's interesting. is a really fierce debate, and people scream at each other at meetings, and it has, in some ways, it's kind of stuck. It's also helped disparities research has become institutionalized in a way that's really problematic. So what needs to come in, nobody wants to talk about this piece, but how do careers come in and sustain this? And we're all implicated in that right now. So I think what did, really this started with women's activism, with the women's health movement of the 60s and the 70s. And the women had, they were often upper class, and they had a lot of resources, and they worked Washington. And then minorities kind of got tacked onto it. And Stephen Epstein sort of, what women have got now through that kind of research is a lot of research on their weird biologies. And the same thing is playing out. So there's a lot of concern. And then there's a lot of feeling that how do you get out of it? Because NIH does require, even years ago when I was doing basic laboratory research on cells, cervical cancer cells, I was supposed to designate race. But this doesn't make any sense for the research I'm doing, but you still have to. So, I don't know if I'm answering your question, but this is not resolved. It's a very hot debate right now.
Other questions? Okay, I think thanks, Lundy, for that. Our next speaker is Raj Ramesar, who needs no introduction, but for those of you who don't know Raj, he is the head of human genetics in the faculty, and Raj's interest focuses largely in using technologies for human genomics to understand predisposition to disease in our local populations and translating this information for clinical use. So it's actually a nice follow-up in terms of Lundy's presentation. And Raj's work to date has been in the area of cancer genetics, genetics of disabilities related to blindness, deafness, and intellectual disability, and psychiatry.
Highlight right now that I'm purposely presenting to you the technological aspects of genetics in terms of the level of resolution one can find in DNA as a science or as a technology. And that's what I'd like to get across to you. And I'm hoping that out of the discussion is going to emerge what's the responsible use of this technology. Okay? And it's embedded in transdisciplinarity and how we work with other disciplines, and particularly the humanities. So the subtext to what is the nature of human genetic variation, and I'd like for this to be understood in the context of ethical, legal, and social implications, which has been a constant part of the Human Genome Project since its inception. Its acronym ELSI. All right, so the interest in the Human Genome Project actually stemmed from noticing that there were certain disorders that ran through families, or ran through even communities. In this instance, I've used the archetypal pedigree or family tree of Queen Victoria here on the top, and how that family or the extended family was driven by haemophilia in the royal houses of Europe. And something that I present to medical students in first year, and this is oversimplistic, is that you are at greatest risk of dying of diseases that affect your parents. In brackets, I've got uncles, aunts, and siblings also. And that's an easy message to get across in terms of both the importance of genetics and the environment. So the Human Genome Project was born out of these inherited disorders in the first instance, but it has its implications are now obviously in many aspects of life. Our interest is primarily in its use, the use of genomic tools in understanding biology that underlies diseases. So the Human Genome Project is one of those multinational projects, but based mostly in the United States and in Europe. and that has yielded the complete sequence of human DNA. Now, straight up front, I want to put these are not my words, but something that came out in this particular publication, that there is no trace of race that has come out of the Human Genome Project. So there's nothing, no information in the Human Genome Project or in the Human Genome that pertains to race. However, I'm going to emphasize the level of genetic variation that one sees across human beings generally. So, what has come out of the Human Genome Project, in fact, emphasizes what we've known through Darwin and others in that genre, is that we can actually map human beings through evolutionary history all the way through their relatedness to all of the other species of life on this planet. So again, I'm stating it oversimplistically. Genetics argues the oneness of life forms based on just 4 letters, 4 chemical letters, A, C, G, T. These are the base pairs that constitute our DNA, and we call that the common code of life. Human beings are not unique in any particular way. I know certain disciplines will argue, of course they are, but we see them as a continuum of life forms. We, in the sciences, and certainly in the health sciences, appreciate our relatedness to other organisms, so we gladly look at either the behavioral aspects of other animals or, in fact, the genomes of most of the lower order organisms in terms of understanding the biology that might imply, or that we share with these other organisms to understand behavior and disease. And importantly, what we do realize is that for all of this evolutionary process, up to humans here, is that there has been a process of adaptation and selection from simple organisms to those that you would consider most complex. So I give this as a quiz to medical students also as to just give me an idea, if we're related to all other species, what percentage do you think we're related to all of these others? And answers here already, so you don't need to strain your brain. However, the last one is, maybe you want to guess, how similar are we as human beings to chimps? Just anybody, stick your hand up and give me an answer. Very good. Some people more than others, obviously. So, and that was a picture of some specific person. So, and again, this is just a slide for perspective. If we plotted all of life on the planet into a 24-hour scale, if life began there and came around all through here, just as a matter of interest, human beings, modern human beings, Homo sapiens, appeared about 2 seconds before midnight. Okay? And all of recorded history is in about a quarter second before midnight. So I mean, just in terms of scale, because we're spending all our life talking about how similar or how different are we as human beings. Okay? In the scale of life, it's actually minute. Unfortunately, or fortunately, we have the capability of science, and as much as when we're thinking about astronomy, we have access to tools or instruments such as these fancy, remarkable telescopes that get us to look so far out that we're able to look far back towards the origins of the universe. Similarly, on a scale of looking into our biology, microscopically, or I don't know if this word nanoscopically has ever been used, but whatever. So we have the capability of using technologies to look into our cells, all the way into our DNA, and be able to map the origins of life even in our DNAs. I mean, that's a remarkable concept, but the information lies there in terms of tracking our history and showing us what other species we share biological material with or our biologies with. So, if again, let's just go back to human beings, and this is the Wisteria-Vine view of human evolution and diversification. This, by the way, is a common sense illustration of how all life forms may be related one to the other, all the way to a root or the origin of life. But this is a diagrammatic representation by our colleague Ken Kidd at Yale University in terms of humanity, that the most diverse aspect of humanity is embedded here on the continent of Africa. Humanity arose in Africa, expanded considerably on the continent, and obviously there were offshoots from the African continent that have populated the rest of the planet. That was a Wisteria view. This is Smarty's view of the world. Again, what I've illustrated here using color is the level of genetic variation present on the African continent. And I just wanted to highlight, and again, this is Ken Kidd's slide, of only a subset of the variants in Africa migrated off the African continent, and you would see the subset diminish in variety as they populated both ends, the tip of Southern Africa, Southern America, for example, or all the way down to the Antipodes. Now, this is where I'm just going to highlight some of the technologies that we use in genetics. This will be the subject of discussion, obviously, this is a principal component analysis that looks at, and I want to emphasize that we as human beings are anything between 99.8 to 99.9% identical, as some of you would already have mentioned, genetically, at the genetic level. The geneticists, and most of us in the audience, may be interested in, you don't often want to figure out why your brother or sister looks so much like you. You're taking that for granted. And in terms of the role of health science workers, for example, our interest is in what's different about us that predisposes us to disease. Or why would the people, 10 people living in a little home, react differently if the flu bug were thrown into that room or TB bug was thrown into that room? Not everybody in that room will develop TB or flu, for example. So our interest is in looking at the genetic differences between us, and that's what I'm highlighting in terms of the technologies that reflect potentially the differences in our biology. Okay, so we have those technologies accessible to us, and this is whole genome sequencing, for example, that's available at the moment, or in fact something like the slide I showed initially with those various multicolored markers. You've heard the word SNP used by Lundy earlier on. Is a single nucleotide variant of the genome, and on a little slide the size of my thumbnail, you would be able to imprint probably, or you would interrogate anything between 1 million to 2.5 million points of the genome as a proxy of that person's whole genome. Okay, but you won't be looking at those bits of our genome that are most identical or similar. You would be attempting to identify, or you would be surveying the most different aspects of our genome. So again, I'm highlighting that we look at the most variant aspects. That's 0.01% of our genome is interrogated on those SNP chips. Using that information, and again, you could query as to why this is done. But what you're able to see here is a sampling of the European population, different nationals of the European population, and this is a principal component analysis of these populations where in 2 dimensions you plot the genetic differences between them, all right? With the most similar and the most different, okay, of these, let's say, 1 million markers, how they align. And what you're able to see is the distinction here, and you will see there's a confluence from this end to the other. There isn't a clear demarcation of almost like a rainbow, all right, that you're able to plot here. But you are, and you would be forgiven for seeing the map of Europe in that principal component analysis. So this is the map of Europe using the same data I've shown you previously, and you can see these individuals fall in mostly into the countries from which they said that they had originated. I mean, this is the power of the technology. We need to think about how it could be utilized. Again, I don't want to abscond any responsibility. We've obviously done this kind of work in South Africa, in Southern Africa, using some of the populations that I've got illustrated here. And what I want to simply highlight is, again, looking at these differences, this is a principal component analysis again. And what I'm highlighting is this is a Japanese-Chinese population, again, using these 1 million variant markers. This is a Caucasian population, and here's an African population, our Southern African population. And you can see they do separate out, again, based on these differences. Again, what I want to highlight is, what are you looking for? So that's because we've designated these groups here, and what you will